So this is really exciting stuff, and we're going to take you now to another very interesting area. Dr. Tim Yu is online, and he is at Boston Children's Hospital, and he shot to fame with the New York Times, and I think it's Myla's story, not Miller. Is that right, Anne? Where's Anne? Miller. Sorry, I got the wrong way around. And Tim is going to tell us about N-of-1 trials and their place in the rare disease trial ecosystem. Tim, it's lovely to see you. Can we hear you speak?
All right, I can go ahead and start. So it's a pleasure to be here. I really had intended to coming to Nashville in person, but family circumstances had required a change in plans. I wanted to speak to you all today about the role of N-of-1s in the rare disease trial ecosystem. And this is a topic that I think is really important to discuss, especially in a group like this. I should say that this is very much an evolving area. I could add a parentheses to this title, say that what I'm going to be discussing are my opinions about this in 2022, and these may change in the future. But the way that I like to approach this is by illustrating through examples the types of N-of-1 trials that are underway and the types of considerations that have gone into the selection of these particular projects and what makes for really a carefully considered and appropriate use of this type of paradigm and what it might instruct us about certain diseases in certain situations. All right, fair enough. And I should say that, again, my slides are really meant to decorate or illustrate a few points, but I'm really looking forward to the discussion, the question and answer session afterwards to get more into some of these important questions. First, my mandatory disclosure slide. So you've just heard from Ed about this amazing technology, antisense oligonucleotides, advanced by Adrian Craner, Isabel, others, Ed's team, and many others, folks at Ionis, other academics, and chemists in the decades preceding. So this technology requires a little introduction. Just the highlights on this slide are just the reminder that these are short snippets of synthetic DNA or RNA that have proven over decades of hard work to become very efficient to manufacture and with appropriate chemical modifications can be delivered, especially for central nervous system applications, quite effectively via intrathecal delivery by spinal tap, or excitingly in the hopefully near to come future by intracerebroventricular injection directly into the brain ventricles. I would like to highlight that in addition to the Tango approach that Ed mentioned, that's really showing wonderful promise for Dravet, there are two general modes of activity for ASOs. They can be used in general to boost expression like in Tango by altering patterns of gene splicing. They can also be used to knock down gene targets, which is especially useful in situations where there are mutations causing toxic gain of function conditions. And folks need likely little reminder of how many of us first heard about antisense oligonucleotides. For many folks in the room, it was this 2018 paper about an antisense oligonucleotide for spinal muscular atrophy showing remarkable clinical benefits for patients with that severe neurodegenerative condition that really put this technology on the map. And as Ed's previous talk and Mike alluded to as well, there are many, many exciting investigational ASO trials underway today in 2022 for diseases that we care very much about, the ALS, Huntington's, Angelman's, Dravet, and really exciting opportunities that are ramping up for SYNGAP as well. Now, to get to N-of-1s, of course, I suspect that many people have heard of this case, which has gotten more than its share of attention in the press. Our own Boston Children's experience with our team and many collaborators at our institution, including Anne, it really began with this case that we first presented publicly at American Society for Human Genetics in 2018 and published in the New England Journal in 2019. This was this case in which we designed this antisense oligonucleotide drug with the sequence shown in green that was explicitly manufactured to address a mutation that we had encountered in a patient with a very rare and orphan neurodegenerative condition, CLN7 Batten disease. And this effort to design a drug for this patient showed really the power and the flexibility of antisense oligonucleotides and the short timeframes that are achievable with this really remarkable technology. And to remind folks, the basis for this drug development effort was the discovery of an unusual mutation in a child with Batten disease that disrupted the pattern of gene splicing. This child had a retrotransposon insertion deep within the 6th intron of her CLN7 gene. That misplaced segment of genetic code was creating a new splice site, and it allowed us to envision tailoring an antisense oligonucleotide drug to block the usage of that splice site. By tailoring that drug sequence to her mutation and then screening using patient cells, we were able to identify investigational candidates that were capable of correcting the splicing defect that she had unluckily inherited. And with a really wide-ranging collaborative effort with input from the FDA, industry, many scientists, we were able to use manufacturing and animal toxicology schemes that were traditionally would take years under a traditional biotech and pharma basis. But we were able to compress that time frame under the clinical circumstances to get through those tasks in a matter of 3 or 4 months, given the clinical urgency for our particular case. And all in all, the timeline from initial patient encounter to being able to deliver an experimental medicine like this was just a little bit over 12 months. And that publication really opened our eyes and many other people's eyes to the possibilities for what one could do if this type of effort could be made to scale. This Mealison effort was the first example of a individualized antisense oligonucleotide. And it has inspired us to work on additional cases, including this one here. This is our second Boston Children's case of a young infant with ataxia-telangiectasia. This infant was accidentally diagnosed with a fatal genetic disorder near birth. She had an abnormal newborn screen that was concerning for a severe combined immunodeficiency. It turned out that she did not have a severe combined immunodeficiency, as worked up by Dr. Jennifer Puck, an expert immunologist and geneticist at UC San Francisco. But she eventually was diagnosed by David Lewis, John Day, and Audrey Foster Barber with a progressive cerebellar neurodegenerative disorder called ataxia-telangiectasia. This is an unlucky disorder to pick up on newborn screening because there are no treatments for it, and its prognosis is very poor. It's fatal by young adulthood, and by age 10, children with this disorder have severe incoordination and deficits, functional deficits therein that are traceable to recessive mutations in a gene called ATM. ATM encodes a critical part of the DNA damage response that is essential for neuronal survival. And this really otherwise grim diagnosis to offer to a several-month-old was only balanced by the fact that we were lucky enough to meet her at approximately a little under age 1. And we were able to discern that one of her ATM mutations was actually very much like the mutation that we had seen in our original Batten patient. It was a mutation that created a new splice site. And like in our Batten patient, it created an opportunity to design an investigational intervention to try to correct it. This slide here shows the mutation that was found in this infant. It was a C7865 C to T change, a single letter change. That single letter change was sufficient to create a new splice donor site in the middle of exon 53. And the use of that new splice site actually created an out-of-frame transcript, led to completely non-functional protein, and was the basis for her severe disorder. It, however, also allowed us to use the same tricks as before for mucin to design a mutation-targeted antisense oligonucleotide to block this missplicing. And again, using patient cells, we were able to screen those candidates and find one that could restore normal splicing and also restore cellular health, looking at the biological downstream consequences of the normal activation of this ATM gene. We were able to show that drug treatment of patient cells could rescue the DNA damage response, as shown by this green immunostaining of two critical downstream components of the ATM pathway, P53 and PCAP1. The molecular biology details, though, aren't really what the focus of our discussion, I'd like our discussion to be. Where we'd like to really spend our time discussing is the fact that this constellation of genetic features plus the clinical severity of her condition, the fact that there were no other treatments available, and the fact that she was going to suffer irreversible neurologic injury without an attempted experimental intervention. Those factors were considered by us and the FDA, as well as all of the other appropriate bodies like our IRB, and deemed sufficient to initiate an interventional trial for this patient at age 2. We would have loved to get there even sooner. It took us 18 months from the time that we first met her to the time that this drug was ready for injection. But still, we were pleased to get in at an age when she had only minimal symptoms. And to date, 3 years later after initiation of this trial, I'm pleased to report that our patient is doing quite well. The drug appears to have been well tolerated from a clinical standpoint. And from a neurologic standpoint, she's age 5.5 right now. And compared to other children with ataxia-telangiectasia, she's at least on the mild end of the spectrum. We've been rather impressed that her clinical ratings and her impressions of her coordination have been actually quite stable over this time. Her brain volume scans, serial scans, have shown relative stability as well. And other biomarkers such as serum neurofilament levels, which are elevated in patients with AT, have remained approximately stable too. It's much too soon to declare success, but I present this not as an example of success or failure, but just as an example of the clinical thinking that goes into this particular case selection. So in thinking narrowly about this space of N of ones, the question that's been driving a lot of the discussion in our group, in many other groups that are working in this space, is, can this type of effort scale? And in particular, might this be one of the approaches towards tackling this long tail problem of genetic disease with a large diversity of different conditions numbering in the thousands, many of them very, very serious, fatal, or life-threatening, and too many of them too rare for traditional commercial interest. Can lessons be learned from these efforts to leverage efficiencies, to streamline the drug development process, to compile data across multiple efforts, to learn from successes and failures, and to generate efforts that might help patients and additionally provide critical clinical tests of therapeutic hypotheses to find out if there's a therapeutic window that's open in a particular case, to road test a particular clinical biomarker that might actually help set the stage for clinical trial readiness for future ASO trials or perhaps non-ASO trials, even gene therapy approaches or small molecules. And so that's what's driving a lot of the interest here across the field. But that doesn't necessarily mean that an N of 1 type approach is appropriate for any given particular case. And in particular, we have to be pretty cautious still in the exercise of this type of opportunity and the pursuit of this type of opportunity. And the concept, while perhaps deceptively simple, still is associated with many hazards that have to be navigated. Some of these are purely scientific or clinical with respect to the health of the patient and the risks of the trial. Others are subtler but also critically important, like regulatory risk or ethical and justice concerns about patient selection. And anytime that interventions are proposed in a small group of individuals, one has to pay attention to the criteria in which from which those individuals are being selected to make sure that they're really fairly applied, so that one doesn't fragment a population that already is swimming upstream because the condition is not especially common. With all of these points, we really do try to stress that we have to be very careful in managing our own as well as the community's expectations about this, and always be thinking about balancing these disease and treatment risks. That allows me to offer my first comment about sort of generalizable lessons for where the niche is for N of one or N of small interventions. To date, the cases that have been advanced in this space, as illustrated by the two examples I described, and others from Columbia and University of Massachusetts, and several others being put forth by NLORM, they have, to hopefully strong degrees, really captured these three critical considerations. First is the demonstration of a really compelling and time-sensitive medical need. To date, these interventions have been advanced in genetic conditions that are fatal, generally progressive, where there aren't other options, where the natural history really balances the risks of treatment against the risks of no treatment, the risks of the disease itself. And critically, the time factor is really important in that consideration as well. There are diseases that are awful, but they may not necessarily progress, or they may be awful for some period of time that's stable. And the question of whether you should intervene now, or whether one should invest additional effort and intervene in 1 year, in 3 years, or in 5 years, these are not easy questions, but they have to be considerations for any patient population. Point number 2 on the slide. All of the types of N of 1 interventions that have been pursued to date have involved, unsurprisingly, single gene disorders. These are Mendelian monogenic conditions, so-called simple Mendelian conditions, where there's a particular identified mutation for which there's a scientifically feasible strategy. Now, that strategy might vary slightly from case to case. Sometimes they are like Spinraza, where splice modulation is sufficient to boost levels of a critical SMN gene, or like in CLN7 bat disease, where again, splice modulation is key. Other times, one may be looking at variations on that theme, like Tango, or other types of mechanisms like knockdown, like ASO knockdown, and I'll provide an example of that. Each of these mechanisms that are engaged changes the risk profile somewhat. And there are known unknowns and unknown unknowns associated with these various approaches. And so each of them requires some nuance in thinking about it. It's not a one size fit all. If it's an ASO, therefore you can treat it exactly the same. I think the particular mechanism that you're engaging makes a big difference. Lastly, this last point, a unique mutation found in one or very few patients. In the case of the Mielsen effort, our initial effort, one of the standout properties of that case that allowed us and the FDA to proceed was that the mutation that we were addressing in our patient was actually found only in her. We have not yet found this particular mutation in anyone other than her family, and other individuals in her family were just carriers, so therefore they did not need intervention. And a similar story applies to the ataxia-telangiectasia case that I described, at the time that we initiated our trial for our AT patient, she was the only patient in the world that we knew of with that particular mutation. Interestingly, since that initial trial, 6 other patients have now surfaced. This is a recurrent mutation, and it's not a true N of 1. However, 6 or 7, counting the pilot patient, is still a number which is sufficiently small that the FDA has allowed us to proceed to enroll an additional second patient, and we're working with authorities to try to enroll as many of the others as we can. And the theme here, the point here, is that under these circumstances of very small numbers of patients, one can carefully and individually consent these efforts, family by family. And the intent here is not to seek a approval from the FDA to broadly market a drug for registration and approval, but to recruit a small number of individuals with ethically justifiable inclusion criteria for which these efforts can be applied. So those are our considerations, and those considerations have been essentially reflected in the draft FDA guidance, which now has a series of guidances that were released last year that offer the standards for when the FDA will consider an approach like this. If you meet these criteria for situations involving ultra-rare, severely debilitating, or life-threatening conditions, if you're using an antisense oligonucleotide of known chemistry, then the agency is willing to offer accelerated reviews and streamlined requirements, for instance, single species toxicology requirements that might allow you to avoid time-consuming and expensive animal studies, for instance, in non-human primates, as an example. So that is reflective of the considerations that I mentioned. I'd like to close with one final example to drive, I think, home the point for why these considerations are so important. And this is an example that I'm pulling out of our own experience, recently described and recently covered in an article in the New York Times that illustrates a cautionary tale for, again, how important it is to be very careful about when one takes these types of risks with these accelerated approaches. And this case, I know, has also been the subject of a lot of discussion in the community. I'll present it here with a proband, a 9-month-old with a condition called KCNT1 neonatal epileptic encephalopathy. This infant had a clinical syndrome called epilepsy of infancy with migrating focal seizures. On day of life 2, she began having dozens of seizures per day that were refractory to medical treatment. Eventually, she was found to have a de novo mutation, an R474H mutation in a gene called KCNT1. KCNT1 is a neuronal potassium channel, and like the gene target in Dravet syndrome, this is a potassium channel which is critical to regulating the excitability of neurons, such that mutations in this gene that cause a toxic gain-of-function effect that create leaky channels lead to striking neuronal hyperexcitability and these refractory migrating seizures that are so clinically and electrographically characteristic. As illustrative of the severity of this disorder, this particular index case, when evaluated at 2 years of age at Boston Children's, slept most of the day. Her parents reported she would open her eyes but wouldn't track visually. She could slightly lift her head when placed in a prone position, but she exhibited no purposeful movements. She could feed orally, but had choking concerns, and she was having approximately 20 seizures per day. Unfortunately, this clinical snapshot of this patient is entirely typical for patients with KCNT1 mutations and this clinical syndrome of EIMFS. In these 2 case series published a few years ago of 135 cases and 17 cases respectively, 100%, nearly 100%, well, actually 100% of all of these patients suffered severe to profound neurologic impairment if they had the combination of mutations in this gene and this clinical presentation. In particular, in this second case series of 17 patients for which detailed records were obtained, 8 of 17 passed away, often from SUDEP. And when they did pass away early, they passed away at a median age of just 3 years. As I mentioned, R474H is a known bad actor in this field. R47H is a mutation that's been characterized quite well. In fact, it's the strongest of the known KCNT1 mutations, and it is the mutation that was encountered in this index case. Mutations in this particular mutation is associated with a 25-fold increase in potassium current transduced through the channel. And that increase in potassium current leads to an increase in the amplitude and number of action potentials that are generated by neurons with this mutation. And these neurons also fire with a shorter interspike interval, indicative of a diminished refractory period that again illustrates how excitable they are and how devastating and how difficult the seizures are to control. And this recognition of a clear toxic gain of function mutation as the mechanism led us to pursue an ASO strategy, a very simple strategy, to knock down the toxic gene. This is not an upregulation strategy like in Dravet. This is a knockdown of a toxic product which is wreaking havoc on the set point for firing for neurons. And so we can use specialized antisense oligonucleotides that activate RNase H to specifically bind to the KCNT1 gene, cleave it, and reduce levels. And compressing a huge amount of work from multiple institutions and multiple parties, we screened hundreds of ASOs and found several candidates that had the properties that we wanted, that they could knock down KCNT1 gene in patient cell lines, in mice, and could even rescue a mouse KCNT1 epilepsy model. Work done in collaboration with Steve Petru's lab showed this beautifully, and also extended into another mouse model with Len Kaczmarek at Yale. And this preclinical data package, combined with GMP manufacturing and animal safety studies in mice and intrathecal injections into rats, was submitted to the FDA and was the basis for being granted permission to enroll a very small number of patients in September 2020. In particular, we enrolled 2 patients with this most severe and well-known R474H mutation, who presented with this very severe phenotype of epilepsy in infancy with migrating focal seizures. And Dr. Christelle Ashkar, my colleague and work partner in all of these trials, assembled a clinical protocol with a careful intrapatient dose escalation with many safety and efficacy evaluations scheduled at 4 to 8 week intervals. How did we do? I'm just going to jump to show you the seizure responses that were seen in this investigational effort. Here is a track of seizures per day recorded by the family of patient number 1. This patient was having, prior to investigational therapy, between 5 and 30 seizures per day. And then in the red stars, you see ASO doses given at 10, 20, 30, and 40 milligrams, and then spaced out at wider intervals. And what you see is that there were significant reductions in seizure count and severity over the first year of the trial. There is a change in the color scheme around day 250, which is a little bit disconcerting. But there is the development of new myoclonic seizures at that standpoint that forced us to change our scoring system. So apologies for that discontinuity, but overall between these types of reports, as well as 24-hour EEG quantitative measurements, we were convinced that we were seeing substantial reductions in seizure count and severity, although they certainly did not go to zero. Case number 2 showed a result which is, I think, easier to talk through. Case number 2, this patient was having between 5 and 20 seizures per day before the onset of the trial. And then she received doses of 10, 20, 30, and 40 milligrams. And what you see is that after the 40 milligram dose, we saw a really striking reduction in seizures, where seizures came down from approximately 10 to 15 per day to between 0 and 3. And they stayed down for nearly 40 days. This was a really striking response that I think is evident without any statistical analysis to folks in the room, and it was corroborated by 24-hour video EEGs as a objective quantitative measurement too. What you also see in this graph is that after day 100, as further doses were held, that seizures frequencies gradually returned. There is an on and then off response as the seizures came back over a slow time course, suggestive of a half-life of the drug of something around 8 weeks. And this type of on-off response is very convincing for an on-target effect, impacting seizures in the way that we had hoped. What I haven't explained, though, is why doses were held after the 40 milligram dose. And the 40 milligram dose in this patient was her last dose for quite some time, because as we reported at the American Neurologic Association in Chicago a month ago, and was also covered in the New York Times in an article around the same time, that we encountered serious adverse events in both patients that led us to pause the trial and to halt dosing, at least temporarily. In case number 1, at the end of the first year, we did an MRI, which actually led to a rather surprising finding of severe hydrocephalus. We had not seen clinical indications of worsening during this time. In fact, her family had reported that she had been doing quite well. But so the MRI came as quite a surprise. And this finding at the end of year 1 led us to pause dosing, pause the trial, consider clinical interventional options. After really careful and nuanced discussions with the family about her quality of life, this led to a decision for the family to withdraw from the trial and a transition to comfort measures. She ended up passing away a few weeks later. In patient number 2, ventricular enlargement was observed shortly after the 4th dose. This was caught at a much earlier stage, and this, in discussions with the family, was treated with a VP shunt to relieve the potential effects of worsening enlargement. And VP shunt placement corrected this problem, stabilized her clinical condition, and allowed her to be discharged from the hospital a few weeks later. Of note, we have, in discussion with other folks in the field, discovered that there's additional evidence of dose-dependent ventricular enlargement seen in a Huntington's ASO trial in a group of 192 patients in this Ionis and Roche-sponsored trial. And so we now think that intrathecal ASOs, some sequences at high doses, might incur some risk of this type of ventricular change. This might be a matter of too much too fast. It's too soon to know for sure, but this is an example of the types of new findings that we have to be on the sharp lookout for as we proceed in the space. In the case of this particular child, weighing the risks that we incurred, plus her natural history of disease, we are, of note, planning a dose reduction and resumption, and have received approval from the IRB and the FDA and the family to pursue that in the next few weeks. So I think that last case, I present it in the spirit of illustrating why these considerations, I think, are so important. Why, as we think about ASOs and these types of trials and the risks that we take on, and we think about our patient populations, we really think about this in a balanced way, that focusing on these types of really fatal and orphan conditions in small numbers of patients is right now the right thing to do as we work out how best to use and take advantage of this wonderful scientific tool, while still paying a close attention to the clinical risks that we'll only learn about through interventions and treating patients. Thank you so much. I look forward to the Q&A time, and let me stop right here.
Thanks, Tim. That was fascinating and measured. It was both a wonderful talk. I think we've gone a bit over time. We'll take one question now, and because I don't want to shortchange Dr. Collar. There was a question here. Was there a hand in the air? No? I think it was one of our... Anybody with a question? Yes, over here next to Dr. Paduri.
Hi. When you say that the FDA is looking at, N of one or N of a few, I know you mentioned that 6 patients was something that they approved with the AT patients. Do you have any sense of what's the limit of a few? Like, is it 10? Is it 20? Is it 50? Thank you.
That's a really good question. And I would say that on paper, the guidances actually allow, they state that these guidances are expected to apply to situations where there are no more than 2 patients. So actually they haven't on paper documented 6. I would think that at some point, the question is how many patients might be allowed into the trial. There's also questions about, well, what are the appropriate criteria by which you can select those patients? If it's selecting 2 or 6 out of a pool of 50, that actually raises really important questions about ethical inclusion standards that can be equally applied to patients too. So for the moment, there's a lot of gray area right now around how one gets from 2 to these smaller numbers. Let's take that on in more detail in the later Q&A.